Good afternoon and welcome to the Harvard Radcliffe Institute. My name is Ido Berger. I'm faculty co-director of the science program at Harvard Radcliffe and professor of astronomy at the Harvard Faculty of Arts and Sciences. It gives me great pleasure to welcome you to our annual Next in Science Symposium, this year focused on spotlighting women's health and associated challenges and disparities. This is a hybrid program, so it is great to see so many people here in the audience and to welcome those joining us virtually from all over the world. The Next in Science series is one of my favorites here at Radcliffe because it highlights the work of energetic early career scientists whose creative, cross-disciplinary, and cutting-edge research is thematically linked and stands to transform their fields. Today, we gather to illuminate and address one of the most pressing challenges in contemporary healthcare: the historical biases that have pervaded clinical and translational research, often resulting in misdiagnosis, ineffective treatment, and overall inadequate medical care for women. For most of the 20th century, medical research was characterized by the exclusion or underrepresentation of women in clinical studies. The National Institutes of Health Revitalization Act of 1993, that's right, 1993, was a crucial turning point, mandating for the first time the inclusion of women and minorities in NIH-based clinical trials. The use of female mice in research to model biological sex as a study variable was not required until less than a decade ago, in 2016. The 1993 legislative milestone has undoubtedly shone a light on the gender disparities in healthcare outcomes. Yet we find ourselves at a juncture where serious inequities demand our urgent attention and action. Research on women's health remains underfunded. According to McKinsey, in 2020, just 4% of all biopharmaceutical research and development spending went toward female-specific cancers, and just 1%, less than $2 billion out of $198 billion, was allocated toward female-specific conditions. Such underinvestment creates wide gaps in our understanding of conditions that are specific to women, predominantly affect women, or affect women differently. These include not only conditions that predominantly affect women, such as breast, cervical, ovarian, and uterine cancers, but also broad disease categories, such as cardiovascular disease, where underdiagnosis, misdiagnosis, and delays in treatment lead to men being 23% more likely to survive a cardiac event than women. The pathogenesis of dementia in women is also different than in men's, in ways that we are only now beginning to understand. Today, we want to explore and redefine the frontiers of women's health. Through cutting-edge research, innovative policymaking, and transformative breakthroughs in public health, mental health, diagnosis, treatment, and drug discovery, we are charting a course towards a future where women's healthcare is as comprehensive, effective, and nuanced as it must be. We are joined today by some of the most inspiring minds in the field who are leading this charge with bold ideas and groundbreaking projects. Together, we will explore how these advancements are not only addressing critical needs, but also paving the way for improved health outcomes for women everywhere. Let us engage in meaningful dialogue to challenge the status quo and to collectively envision a healthcare landscape where gender inequity is a relic of the past. Before I introduce today's speakers, I would like to provide a quick orientation to our program. The program will have 2 sessions with 2 speakers in each session, separated by a 10-minute break. For those joining us online, the live stream will continue through the break, so there is no need to sign off and back on again. Each speaker will offer their presentation, which will be followed by a question or two from my colleague and co-director, Dr. Immaculata De Vivo. Dr. De Vivo is the Melanie Mason Nemec 71 faculty co-director of the sciences at the Harvard Radcliffe Institute, professor of medicine at the Harvard Medical School, and professor of epidemiology at the Harvard T.H. Chan School of Public Health. After the individual questions or presentations, we will have a group discussion and audience Q&A moderated by Dr. De Vivo, which will conclude at about 4:30 p.m. Audience members, whether you are here in person or online, are welcome to submit questions at any time during the program and the individual presentations using the Slido link provided on the screen behind me and posted in the chat of the Zoom webinar. We will try to get to as many of the questions as possible during the Q&A session. I want to take a moment to acknowledge the members of the Radcliffe Institute Leadership Society and our annual donors who are watching this afternoon. Your generosity keeps Radcliffe programming free and open to the public, and we thank you. We also want to gratefully acknowledge the Melanie Mason and David W. Nemick Fund for Science, which is supporting this event. And now to the speakers of the first session. Our first speaker is Dr. Tara Castellano, Assistant Professor Clinical at the Louisiana State University Health Sciences Center, Department of Obstetrics and Gynecology. Dr. Castellano's talk title is "It's Still a Man's World, Doing More with Less in Women's Health Research." Our second speaker of the session is Dr. Victoria Brown, Biology and Drug Discovery Consultant at Versant Ventures. Dr. Brown's talk title is "The Case for Precision Medicine in Ovarian Cancer." It's now my pleasure to invite Dr. Castellano to the podium.
Thank you so much for the introduction, and it's my privilege here to talk about my work and what I do in women's health research. Some disclosures. So women's health research, why now? I am a GYN oncologist by training. I think it's the best job. I get to meet women at a vulnerable time in their life, diagnosed with cancer. I also get to perform their surgeries and prescribe their chemos and do treatments with them, get into survivorship, into surveillance, and also end of life. So it's a very great job, and the women are the best part of my job. I tell people I came here for the surgery, but I really stayed for the science, and the science in cancer research is really amazing. We have a lot of cutting-edge drugs and surgeries, interventions for patients. So now I spend a lot of my time working as a clinical trialist in GYN oncology. So bringing those cutting-edge drugs and interventions to my patients in South Louisiana is a big part of my fulfillment and my joy, especially on hard days. And I have to be honest with you guys, it's been a lot of hard days lately, I think, when you do this work in women's health and women's research, to feel suddenly like your work is devalued. So I was feeling a little bit of pity for myself, how to come here with optimism and joy like I usually do when I talk about my work that I love, when I think a lot of us are feeling a little bit of uncertainty. But I got over my pity party, and I really think more now than ever it's really important to come at this topic with our enthusiasm and our strength and grit to get ready to continue to do some work. Thank you. I think we know why it's important. Women make up more than half of the population, and as Dr. Berger alluded to, we've been left out for a long time. Pretty recently, we're still left out. Women, I think, were left out initially for some reasons. Initially, just misogyny, understanding that the male body was the norm and women were just smaller versions and we could just apply smaller doses or less surgery. We know that doesn't work. I think another issue is that a lot of women's value is valued around their reproductive health. So there's a lot of gaps that we've gone over here in understanding that it's more than just being a smaller man, but there's a lot of sex and biological and anatomical and physiological differences between sex, gender, and biology. And I think this is a great example. Sally Ride recalls that some of the greatest minds in NASA really thought one woman in space for 1 week needed 100 tampons. So there's a lot that we still need to improve. I think, where did the skewed value come from? Like I said, there's this prejudice that a woman's value is centered around reproductive capacity. And I think the thalidomide crisis can really kind of center this a little bit in some interesting ways. So thalidomide was a drug that was very effective for morning sickness and prescribed pretty widely in the 50s in Europe and the UK. And it had devastating congenital defects, birth defects that affected tens of thousands of families. And I think there were two outcomes that were pretty juxtaposed but important. On one hand, it really showed and highlighted the need for testing for efficacy and safety in drugs and medical interventions, especially in vulnerable populations such as pregnant women. But on the other hand, it kind of reinforced this belief that women should be untouchable. If you do too much stuff, you can, affect their reproduction, affect pregnancy and lactation. And while those are important, probably come from a good place, it really left women out for a long time, actually leading to a 1977 FDA policy which explicitly excluded women of reproductive potential from participating in phase 1 and 2 clinical trials unless they had a life-threatening condition. So you can see where this mandate, maybe in good intention, kind of set us back for some decades. And like Dr. Berger alluded to, it wasn't until 1993 that the NIH addressed and made law a policy to include women and minorities in clinical research, really those phase 3 trials that are going to lead to drug approvals and really affect public health. And that cost wasn't allowed to be an excuse to exclude women and minorities. And a year later, the FDA created an Office of Women's Health by congressional mandate to protect and advance the health of women through policy and science. But is it enough? Well, I think we're here because we still have a lot of work to do. Even through those mandates, we did have some good evidence and data that came through, and the Institute of Medicine in 2001 released some evidence-based report that demonstrated how sex differences really influence biology and physiology and should be studied as a scientific variable in clinical research, where before there wasn't really a policy that this had to be collected. But as recently as 2022, a Harvard Medical School study did find that as of 2019, women are still substantially underrepresented in clinical trials. Cardiovascular disease, where we think of as a male disease, but affects 49% of the people with cardiovascular disease are women, but only 42% participated in clinical trials. Psychiatric disease, where women account for 60% of the patients, only 42% of clinical trial participation. And we see those similar trends across many diseases and cancers like I manage. I'll focus a little bit on federal funding, but I just always want to keep in the context that actually industry funding actually spends more money in R&D towards medical device and drug development annually, but they tend to follow the federal mandates and policy, right, to stay aligned, because at the end it's all about getting things through the FDA. So when we look at the NIH and their institutes of health, you can see out here in orange is the federal funding for women's health research, and this is housed across many institutes. Can be very confusing if you have a women's health initiative. Who's going to help? Whose priorities are going to align with your research? Where do you seek funding? Where do you seek mentorship? Well, the NCI has about 16% of their budget to women's health, and a large percentage of that is breast cancer research, which is important. But the next biggest contributor to women's health research is the NICHD. But even that institute, which has like women's health in the mandate, is still less than 50% of their budget goes to women's health research. So we're already seeing that we're severely underfunded. And we look at the NICHD, or the National Institute of Child Health and Human Development, we see their extramural branches. If we look at the ones that we exclude, pediatric and human development, about half have specific directives to address maternal and women and reproductive health. And when we remove obstetric and pediatric, it's really only 2 or 3 that really are going to address benign gynecologic and female diseases. And we kind of look at the actual numbers at the NICHD. In 2020, the NICHD had a $1.5 million budget, but less than 10% of that went to obstetric, gynecologic, and fertility conditions. For example, endometriosis research was granted $14 million of funding, but only 80% came from the NICHD. Uterine fibroids similarly got $15 million in funding from the NIH, but only less than 0.5% of that came from the NICHD. So if we look at the NIH in general, we see that even though grant funding increased from 2013 to 2023, the proportion actually allocated to women's health has been dropping steadily, 9.7 in 2013 and 7.9 in 2023. So it hasn't been a priority, and there's a lot of work to do. For me, I take care of gynecologic cancers, so when I look at gynecologic cancer funding at the NCI, we know breast cancer is going to get the biggest piece of the pie. It's the most common cancer to affect women. It makes sense. And it's not a zero-sum game. I don't want breast cancer to get more funding, but I want gynecologic cancers, or less funding. I want gynecologic cancers to get more funding. The last line is the numbers for prostate cancer, and you can see here prostate cancer gets a significant amount of funding when you compare to the women's cancers. Now, I am a specific proponent of uterine cancer. There's a lot of disparities in uterine cancer. It's a curable, detectable disease, so research funding could go a lot farther for saving lives. It's historically underfunded, historically impacts Black women, minoritized populations way worse, is more of a lethal disease, similar to prostate cancer. And again, not to take away from the importance of funding prostate cancer is more common than uterine cancer, about 4 times more common, but it is 19 times more funded than uterine cancer. So we see these disparities across the board. And this is a nice schematic just looking at lethality. When we think of the lethality of a cancer, prostate cancer is very well funded when we think about the impact on person years of life lost per 100 cases, whereas all the other gynecologic malignancies fall with certain other disease sites that could use more funding based on their lethality. I believe the picture gets more bleak when you look outside of the cancer world and we're just looking at benign conditions that affect women. This is a good schematic that shows the NIH dollars per affected patient, and this is 2020 data. The blue bars on the left affect the condition and the persons affected, where something like menopause, if we're lucky enough to get to that age, half of the population will be affected by this condition. And essentially, there's $0 per person directed to menopause research. The red line will represent the research per person affected, research dollars per person affected. When you go down the line to other benign gynecologic diseases, fibroids, endometriosis, about $1 to $0, $8 for infertility. We can see the cancers are well funded. That's great. There are deadly diseases. We want those fundings, but we want some more funding for common conditions that affect women. And you can see Alzheimer's disease, Down syndrome, things that are well funded according to their impact. So there's some disparities, there's some areas to improve equity in funding. So I think all of this goes to show why there was a mandate approximately a year ago in March. Then President Joe Biden did sign an executive order to build upon the NIH-wide effort to invest $200 billion in the fiscal year 2025 for interdisciplinary women's health research. And the aims to study the complex intersection of women's health, to include diverse populations in clinical research, as well as a diverse workforce that performs this research. As of a Google search this morning, there's no real clear idea of where this executive order stands now, but a lot of these words have been stripped from NIH funding. So I think there's some troubling times ahead, but the initiative and the mandate, I think, are still really important, and the work still has to be done. So based on that, the National Academies of Sciences, Engineering, and Medicine met in 2025. They are asked to convene and make an assessment of this NIH request to improve women's health, and there are really 4 big take-home messages from that meeting. The first was that there's a significant funding disparity. Less than 8% of the total NIH grant budget for the fiscal year 2023 was allocated to women's research. We talked about that. The need for improved strategic NIH-wide priority setting, oversight, and adherence to policies that support women's health research. Number 3 and number 4, I feel, are the most important. We're going to kind of take to the end of this talk for me, but the need for a specific institute on research. So remember all the institutes. We need an institute of women's research. That's going to be kind of the guiding light. Who's going to oversee these priorities and make sure the follow through and oversight happens. And last, which is really the most kind of prescient to me, is that we need for sufficient training and additional funding to grow and retain women's health research workforce. Who's going to do this work? And I bring that up because I'm here with a lot of amazing scientists, but my route was a little bit different. I don't come from a PhD background or a basic science background. I don't do translational work, but what I do every day as a clinical trialist is I bring that research to the patient at the bedside. I started my track in medical school as a non-traditional student. I came to medical school, that's me and my 2 sons, as a mom already, and it's not surprising that I was drawn to the field of OBGYN because every interface I'd had with the medical field was through a reproductive lens. I was lucky enough to match at an amazing program at UNC Chapel Hill where I was just surrounded by amazing, incredible women's health researchers. We were putting people on clinical trial who were pregnant, getting induced for labor, having pelvic pelvic floor disorders. And I thought this was kind of normal. I was naive. I thought, oh, I guess all OBGYN programs do this. This was actually a very unique kind of setting to get trained. But I'm still getting trained. I'm working 80 hours a week. I have 3 weeks off the entire year. I have kids. If you're learning how to do research, you're doing it on nights and weekends. And that's what you had to do if you want to do a fellowship. So I did it. I trained. I had a lot of mentors that helped train me. I was able to match in my fellowship, which is another 3 years. So already, 10 years into my life. And I'm not going to run a lab, but how can I take the things that I learn? Well, at Oklahoma, at OU, where I did my fellowship, I knew I wanted to go back to Louisiana State University. At OU is it's a poor state. It's on the bottom of the list for a lot of things. But for some reason, they have this amazing top cancer center because they had the right leadership in place. A gynecologic oncologist like myself was the cancer center director. I was surrounded by the top clinical trialists for 3 years, and this really inspired me and gave me a mandate to take everything that I learned there and really bring it back to South Louisiana. So my goal after fellowship was to go back to LSU. In New Orleans, in Louisiana, there is not one NCI-designated cancer center. If I go outside the street here, I could probably throw a rock and hit an NCI-designated cancer center. So the resources aren't there, but the need is there. And it's kind of lofty, but I said, I want to see the balloon drops like I saw at Oklahoma. I want to see that for my patients in Louisiana, my community. And I was very lucky to get to LSU and be surrounded by a team that was kind of primed and ready to go. I had colleagues, I had partners who were already starting a clinical trial program. We had a very robust NCORE, which is an NCI community oncology group that interfaces with community practices. We're the lead site for the Gulf South NCORE. It's a minority NCORE because we have a majority minority population. And I was able to interface, really leverage the NCORE, meet with the local leaders, and really start developing a clinical trial program for gynecologic oncology at LSU. And it took some strategizing. When you're working with less, you have to have some approaches that are novel. So for us at our NCORE, at our LSU site, we developed a virtual research nursing program. We could go into all of that, but basically allows virtual nurses to come into my community clinic sites and enroll patients on clinical trial who might not be able to get to the fancy, shiny downtown hospital. And we know that 80% of patients with cancer are going to get their cancer care at community clinics and not be able to come to the big academic centers and kind of, this middle of a city. And we continue to engage partners. We had a true belief in what we're doing, so we're really kind of constantly building. We've got some attention from the NCI. Jill Biden came to visit our cancer center, talk about our virtual nursing program. We're developing, investigator-initiated trial protocols. And to continue what we're doing locally, we have to continue to engage nationally. So our group is working through mentorship and development. We're serving our national committees. We're helping review and develop research clinical protocols with the NCI, with our cooperative groups, with industry. Making partners locally and nationally is important because all of this comes back to our patient. And that's the part that I think is where we do a lot of our hardest work. Yes, we do a lot of hard work, but the most important job I have is when I'm sitting across the table with my patient. And how do I bring this science that's going to impact my patient population? Well, if you're enrolling on clinical trials, if you're making a clinical trial protocol, portfolio for your site, you have to think of some things. What are your resources? So first thing, I can't open clinical trials that are all phase 1 and have very strict criteria for participation. That's not going to fit my population. I have to have a way that I can get a larger catchment, so virtual research nursing, decentralizing protocols. For example, I will favor opening clinical trials that have, less follow-up. They can get a CT scan. They don't need to drive 200 miles to get a CT scan at my hospital when there's a perfectly good CAT scanner. I don't want those kind of barriers for my patients to participate. Pragmatic design, so you can keep your clinical trial portfolio and budget kind of solvent, right? You can't bankrupt your institution with lofty science. It has to be a little pragmatic. It has to be affordable. But the most important thing is you have to offer participation. So you have to teach yourself to identify your own internal biases, your clinical research team. If you're sitting across from a patient, you can't go in thinking, oh, this woman is never going to want to participate. She has 2 jobs, or she has to drive 100 miles. You just offer it to anyone. If there's a clinical trial that is a patient can enroll on, they meet the criteria, they need to be offered. And then you need to track those metrics. So this kind of spurred us back in 2022. We were doing this hard work, but we said we should really track this and kind of follow it because we're getting some recognition of being pretty successful. So we started a QI project. We were already meeting weekly with our clinical research team, reviewing all new patients, but we started including anyone that was maybe progressing or recurring or ending maintenance. And we were keeping track of the patients we were seeing, so we had a good idea of our catchment, our baseline, and our need. And then we would use that and kind of strategize our resources. And we also kept external measures. There's a lot of things that you can't control for. We had a hurricane this year that was devastating. We had workforce instability after COVID, like many places did. So we're kind of tracking those metrics and what affected clinical trial accrual. And that led to us publishing our data on this experience. And we found that over time, when we kept careful track of what we were doing and the factors affecting this, that our clinical trial enrollment rates increased, and national clinical trial participation is about 8 to 10%. At our site, small but mighty, we have 40% participation. Thank you. I'm proud of that. But I think what we're more proud of is that our participation really goes across racial, ethnic, geographic boundaries. We're going to have the same representation across those things. And that got us offered to do an invited review on clinical trial screening practices. So this is the first step to kind of dismantle some barriers for participation for women on clinical trial. So in general, I'm running out of time, what does a successful clinical trial program look like? Well, it's something that's going to bring the science to the women that need it. This is some of our patient-facing information that we develop for our trials. But you have to be busy. So I'm a busy clinician. I was in clinic yesterday. I'm doing surgeries when I get back to town. If you're not taking care of patients, you're not going to get them to the science. You're not going to have that. So that's the first thing. The next thing, you have to track your clinical trial enrollment. And that's what we found. We have 40%. We've been staying there for 3 years. And kind of, we have some ebbs and flows, and we track why. But you have to have equal participation across racial, ethnic, gender identity, geography. You want to make sure all of that is you're asking every patient. And you have to have robust outreach and community engagement. So a big part of our job is doing things like this, talking about what we do. We never say no to a media spot, but we also have philanthropic events at least twice a year. We have patient appreciation events, because that brings the joy back into what we do. So in conclusion, thank you. This is one of our community events, second lining through City Park. More now than ever, equitable women's health research matters. Even though other people might not feel that way, it matters. Research based in equity, diversity, inclusivity is evidence-based, and it benefits everyone. This isn't a zero-sum game. Clinical trials, clinical research leads to safe and effective drug approvals. If you're not testing drugs and interventions on a diverse population, then you're not going to have safe interventions for everyone. And lastly, maybe this was more for me, but careers in women's health research and equity are still joyful, and we still can find that. Some references, and I just want to thank you and acknowledge my patients, my team, their family, my community, and happy to take any questions.
Hello, my name is Immaculata DeVivo, and I'm the Melanie Nemec Science Co-Director. And just briefly, my colleague Ido described how we were going to do this, but just as a reminder, I will ask Tara a question or two, and then the rest of, I'll introduce the other speaker, we'll have the break, and then we'll have the other two speakers. But at the end, we will have a full discussion, Q&A with in-person audience as well as the online audience. So Tara, thank you so much. That is so inspiring. When do you sleep?
Not very much. No, I do. My kids are older now, so I have that. But yeah, I think if the work's inspiring, it's easier to get out of bed and do it every day.
Well, thank you for coming and bringing the joy and the enthusiasm. So just one question. Like I said, we have more questions later. I have, as you would imagine, so you did enumerate some of the challenges with recruiting into these clinical trials, location, busy, et cetera. But are there some unique barriers to this patient population in southern Louisiana that you encounter when discussing clinical trials?
That's a great question. For Southern Louisiana, some might think it's a barrier, but for me, it's an asset. If you're someone from outside, it might be a barrier if you don't speak kind of the shared cultural identity, if you don't understand. So for me, I'm from there, so I understand, kind of the competing interest of patients and caring for their family. But I think it's a strength to have a community that has a lot of shared cultural identity and values. When you can tap into that, it's easier to gain trust and people will listen to you. But I think it's really about getting trust. And I tell patients, I'm very honest. I say there's a reason you should be distrustful of us because we've not done a good job in the past. So I think that's the biggest barrier is getting trust. I think that's in any community. But I think if, knowing your local culture can sometimes really help you and can be a barrier for some people if you're not paying attention to what's going on outside your hospital doors.
Yeah, I would have assumed that there's distrust in medicine and science in general, but in this particular location, this geography, that has got to be.
Yeah, and I don't find it as much. I think sometimes maybe that's our own internal bias. We think people don't trust us, so we just, it's going to take a lot of time or it's going to take a lot of talking, but people actually want to participate. They want to believe that we're trying to do the best for them.
Well, thank you. I will have more questions for Tara, but I'd like now have you take a seat back in the audience. Thank you. It is now my pleasure to invite, if I may, Victoria to the podium.
Good afternoon, everybody. Thank you to the organizers for inviting me to speak at this nice event. I'm Victoria, and today I'm going to be making the case for precision medicine in ovarian cancer. And hopefully by the end of this talk, you'll agree with me that this therapeutic approach is likely the best avenue for this patient population. Just by way of a little bit of background and to build credibility with you, you can see from the left-hand side of this slide that I am a biologist through and through, and also a local girl. So I got my undergrad degree at Providence College, my master's at Northeastern, and my PhD at Tufts. And you can see from the right-hand side of this slide that I'm an industry scientist through and through. So I've worked at several different companies in the greater Boston area, all with roles of increasing responsibility. And most recently, I am a biology and drug discovery consultant with Versant Ventures. Versant is a venture capital firm that invests in and helps start new companies. The left-hand picture on this slide is my maternal grandmother, Anna, who was born in 1935. The right-hand picture are my identical twin daughters, Evelyn and Camilla, who were born in August of last year. Yeah, thanks. Thank you. I agree, they're very cute. I think they're watching. And so between these two photos represents 4 generations of women. And within this time, very little progress has been made in the treatment of ovarian cancer. And in fact, ovarian cancer remains a leading cause of death among women. It is the deadliest gynecological cancer and the 5th leading cause of cancer-related deaths among women in the United States. Like most cancers, if caught early, the prognosis is much better than if caught late. So if caught early at stage 1, the 5-year survival probability is quite good. It's almost 90%. However, if caught very late at stage 4, that probability drops to 1 in 5, or only 20%. And unfortunately, only 17% of cases are diagnosed early at stage 1. The vast majority of these cases are caught very late at stages 3 and stages 4, at which point the cancer has often metastasized and becomes very difficult to treat. And so why do we have such a problem detecting ovarian cancer at the early stages? Well, if we take a step back and look at the symptoms, bloating, pelvic pain and abdominal pain, feeling full quickly, and urinary symptoms, this is just another Tuesday in being a woman. I had some of these symptoms just last week. And so because of that, women often don't seek medical care until these symptoms become too prolonged or too painful. And by that time, and by the time they receive their diagnosis, It is at that stage 3 or stage 4 of the disease. If we look at treatment for ovarian cancer, the 1970s brought us platinum agents. These agents cause a very specific type of DNA damage that some ovarian cancers and other cancers are very susceptible to. However, all cells in our body have DNA, and because these agents are administered systemically, this damage is in our entire body. And so because of that, these medicines can have undesirable side effects. If we shift forward to the 1990s, that brought us the adoption of taxanes. These agents are anti-mitotic agents, which just means they stop the cell cycle before the cell divides. And this is a great idea for treating cancer, a disease characterized by uncontrolled cell proliferation. But again, we have other proliferating cells in our body, like our skin, our gut, and our blood. And so these medicines also have effects there and can have a very narrow therapeutic window. Despite all of this, these remain standard of care therapy today in 2025. There is one notable exception, and that is in the 2010s, PARP inhibitors became very popular, but these inhibitors work only in a very specific context. And so I'm going to take a small detour to explain how these work and how this is an example of precision medicine. So very simply put, our cells can utilize different repair pathways to address any lesions on our DNA. Just by existing in the environment, we receive an onslaught of DNA damage all the time, every single day, but we can take different avenues to repair that. So if there are any DNA damage experts listening, please forgive me for simplifying it to this extent. But on the left-hand side, we can think that one pathway utilizes BRCA1 or BRCA2 through homologous recombination to repair a lesion. And on the right-hand side, we can call it alternative end joining, and that utilizes PARP. And so in BRCA1 or BRCA2 mutated cancers, like ovarian cancer and some breast cancers, that left-hand side of the pathway is no longer in play. Those cancer cells cannot use that pathway to repair any damage, so they rely completely on the right-hand side. But if we treat then with a PARP inhibitor, we can take out that side, and the cell will accumulate too much damage and won't be able to survive. This concept is known as synthetic lethality and is a very good example of precision medicine. And so this is great. But we know that genomic alterations vary very widely in ovarian cancer. The way that we can visualize this is in this circular plot. Every patient here is represented by a different bar, and as we move inward toward the center of the circle, that same patient is retained. And here we've colored and annotated with symbols different genomic alterations that patients carry. And the patients I just told you about, those BRCA1 and BRCA2 mutant patients, are just a small fraction of the overall ovarian cancer patient population. So the PARP inhibitors will work probably best in these patients, but what about everybody else? Well, I'll draw your attention to this group here, cyclin E amplified patients. Cyclin E amplification occurs in about 20% of high-grade serous ovarian cancer, which is the deadliest form of the disease. Cyclin E amplification also occurs in other tumor types, but we'll focus just on ovarian cancer. So I'll tell you what genomic amplification is, what cyclin E does, and what a therapeutic strategy could be for these patients, and how we could think about that from a drug discovery perspective. So again, very simply put, we know that having 2 copies of a gene is normal, one from mom, one from dad, and most of the time, everything is fine. We don't even think about it. But there are some genes in our genome that are prone to genomic amplification. And this means that there are physically additional copies of that gene in tandem, one after the other, in our genome. And this can drive chromosomal instability. And all that means is that in other regions of our genome then are more prone to breakage, and therefore translocation events, fusion events, mutations, insertions, and deletions. And this can be very tumor promoting. And we know that is the case for cyclin E amplification. But in a normal setting, cyclin E is not an enemy. It is actually an important player in the cell cycle. It's best characterized as a regulator of cyclin-dependent kinase 2, or CDK2. You may recall from high school or college biology that the CDKs are the major regulators of the cell cycle, as outlined here in this diagram. But CDK2, cyclin E, are a special pair. They govern the transition from G1 to S phase, so from the gray part of this cell cycle circle to the purple part. This moment is known as the restriction point, and once a cell passes that restriction point, it commits to a round of DNA replication and cellular division, which are highly energy-intensive processes, and so a cell doesn't want to enter that if it doesn't have the proper resourcing. And that's the job of CDK2 cyclin E. But in the case of cyclin E amplification, we have a lot of cyclin E protein around in the cell, and this can drive really high CDK2 activity. And so if we could think about a therapeutic strategy for cyclin E amplified patients, maybe a good strategy is inhibiting CDK2. If we stop the activity of CDK2, could we halt proliferation only in the cyclin E amplified context? And this would be another example of precision medicine. And so I'm proud to say that at my time at Blueprint Medicines, I was part of the team that developed a selective CDK2 inhibitor that we called Blue 222. It was just after CDK2. And if there are any chemists, this is the structure of the compound as published in our manuscript earlier this year. You may be able to tell that it's an orthosteric kinase inhibitor, which just means it sits at the active site of CDK2 and kills its activity. I should mention that there are other companies that have developed CDK2 inhibitors, but because my personal experience was at Blueprint, that's the one I'm going to be talking about today. And so how can we understand if our hypothesis is correct, that if we treat a cyclin E amplified cell with a CDK2 inhibitor, it would stop growth, but it would have no effect in a non-amplified cell? Well, one thing that we can do in drug discovery is to test our compounds on cancer cell lines in the lab. So this is just in 2D culture, in our incubators, in our tissue culture rooms, and we can add drug and look for whatever effect we're interested in. In this case, it will be cellular growth. And then we can generate a plot like the one here on the right, where if a cell is growing, you'll see that kind of beautiful logarithmic curve, and if a cell is not growing, that curve will be more flat. And so we did this in 3 examples of cyclin E amplified ovarian cancer lines shown here. As we increase the concentration of Blue 222, we see a decreased ability of these cells to proliferate in culture. So with that darkest blue line, which is the highest concentration of 222, you see that flattening of the curve, suggesting that the cells are unable to proliferate when CDK2 is inhibited. So that's awesome. That puts a check mark for us in the bucket that this could be a precision medicine therapy. But what if we treat a non-amplified cell type? So these are 2 other ovarian cancer models that are cyclin E copy number normal. They have other genomic alterations that led to ovarian cancer, as talked about before. Here, the addition of our compound 222 has no effect, or largely no effect. So these cyclin E copy number normal or wild type cells are able to grow in the presence of CDK2 inhibition. So this is looking pretty good. But a large criticism of cell culture is that it's not clinically translatable. And so what people tend to like to see are something called patient-derived xenograft models, or PDX models. These models are made by the surgical excision of a tumor from a patient, and then those cells are regrown in a mouse to grow the tumor back up. And this has several advantages. One, it is direct from a patient, so there should be very little to no genomic or genetic drift. The 3D architecture of the tumor is retained, so any 3D effects will be able to be tested here, and there are some drug targets where that matters. And lastly, the 3D architecture and tumor microenvironment is largely retained. One more thing I'll say is that because this is in a living organism, we can test how our drug gets to the tumor and how the body processes our drug. So this is thought to be more clinically translatable, and we can set up these experiments in largely the same way, but in vivo. So these two models here are from patients who had cyclin E amplified ovarian cancer. Those were developed into PDX models and treated with our inhibitor. And you can see, comparing the vehicle gray line to the blue 222 line, that 222 has robust anti-tumor activity in both of these models, and these models happen to be paclitaxel resistant. And in the cyclin E wild type setting, this mimics what we saw in cell culture, where the addition of 222 has no effect. The vehicle and 222 lines are largely overlapping. There may be some combination benefit with paclitaxel, but that is probably a talk for a different forum. But the take-home message is that 222, or CDK2 inhibition, inhibits cyclin E amplified tumor growth. So if we think back and imagine 4 patients, they may all have ovarian cancer, but they all have different disease drivers. In this example, patient 1 has a BRCA1 mutation, patient 2 has a cyclin E amplification, patient 3 has an ARID1A mutation, and patient 4 has a PIK3CA mutation. And so it makes sense that not one medicine is likely to robustly cover all 4 of these patients. So making a medicine that's tailored to each patient's genomic alteration should have the most therapeutic impact. And this was kind of one of the big aims of doing the Human Genome Project early in the turn of the century. And a lot of companies were made and started off of this concept. But we know that drug discovery is largely a scientific game, but it is also a monetary game. Making drugs costs a lot of money. That's just the reality that we live in. It costs many millions of dollars just to bring a drug to the clinic, and then many millions of more dollars to push a drug through clinical trials and approval. And in strong economic times, this is okay. But in times like today, things are a little bit different. So we know that macroeconomic factors will influence investor and company decisions. Right now, as I'm sure you are aware, interest rates are very high, and this is making lending and raising money challenging, even for biotech. Because of that, investors are a little more risk averse. And so suddenly, a small patient population with a novel therapy is not looking as attractive as treating a chronic disease like inflammation or allergies or weight loss. Right now, that's a sure bet. Think about Ozempic. Half of the country is overweight, and that is a huge patient population. The safety margins on that drug are very, very wide. And so the money is kind of going toward that right now, and there's been a little bit of a turn away from precision medicine. But we haven't cracked the case on ovarian cancer, certainly. We haven't cracked the case on any cancer, really. Patients continue to wait for effective therapies. We want to get away from using these medicines that were invented or identified in the 1970s. It's 2025. I think we can do a little bit better. And so I would encourage companies and investors to not turn away from precision medicine completely. I think there is still a place for it and is likely to have the biggest impact on patient health. We don't want it to take another 4 generations of women to see progress on this disease front. We want Evelyn and Camilla to grow up in a world where ovarian cancer is a thing of the past. And so with that, I would like to thank again the organizers for inviting me to speak at this event, and would like to acknowledge and thank the team at Blueprint Medicines for the development of 222. And with that, I will take any questions.
Victoria, it's always good to remind people of the perspective from industry and how market drives the research, so to speak. So precision medicine is something I know very well, given some of my own research. And yes, this was the promise, one of the many promises of the Human Genome Project. And I had a question specific to that. And my question was, as you showed very elegantly, that there are many genetic roads, mutations to, in this case, ovarian cancer, right? So it's not just one thing that you're after. So can precision medicine work well when a patient has multiple genomic alterations, like you showed?
Yeah, that's a great question, and is a little bit of a struggle in the field at the moment. So if a patient has multiple genomic alterations in their tumor, you could think about a patient that might have cyclin E amplification and 90% of their tumor cells, but maybe in the other 10%, they would have a PIK3CA mutation, for example. And so maybe an investigational CDK2 inhibitor would work really well on 90% of those tumor cells, but you would have outgrowth of the cells that carry a different genomic alteration. And so there, it can still work well, but you'd probably have to do combination treatment, which gets very tricky because these medicines often have pretty narrow safety margins. So when you start to combine therapies, that becomes harder. But this is likely going to be the path forward because we're seeing resistance all the time with precision medicine. And so single agent therapy, probably not gonna work super well. Combinations will likely be what's going to be pursued.
Are combinations the current treatment path in ovarian right now?
So with any investigational therapy, like how the 222 trial was set up, there are combinations with standard of care agents. That's the norm, and I think what is required in setting up a trial. But those standard of care agents are not precision medicine. Right. But there are other trials going on. I can think of some in breast cancer where you have 2 targeted therapies plus another therapy, so sometimes a triplet therapy that are being investigated, so that is ongoing.
Very good, yeah. The standard of care is more traditional still as opposed to the targeted approach. And ovarian cancer is probably no different from breast cancer or uterine cancer, which is the cancer I work in. These are not composite diseases, right? They're 10 different cancers, so to speak, right?
Yeah, exactly. So every cancer is a different disease. And so, that's why companies haven't made the big cancer drug, because there isn't one. It's going to take many, many medicines to help these patients.
Yeah, and that's something I wanted to underscore, because and again, I'm very passionate about this idea of precision medicine, is sort of like this targeted approach. It's never going to be that boutique, because the molecular alterations, the DNA changes that happen at the molecular level are so different, and they run out of the treatment. So there's the resistance that you were talking about. So that's got to be challenging.
Yes, certainly. And like the 4 example patients that I showed, that's going to take 4 different drug discovery teams and efforts to make 4 different drugs and get 4 different drugs through the clinic. And so this is a challenge that we're certainly facing. So hopefully the economy turns around and things can get back to normal sooner than later.
Okay, well, I don't want to take up more of Victoria's time. Thank you very much. Victoria will be back for the fuller discussion and Q&A with everyone. And now I'd like to ask the in-person audience as well as the online audience to take a quick break, and we will be back promptly at 3:10. Thank you.